Okay, friends, so let us begin today's class. Today we will be dealing with a very interesting topic, microbial life. So this particular topic comes under the unit 9, that is evolution and diversity of life forms. So in that particular unit, we will be dealing with evolution and also the different varieties of life forms. And under the diversity of life forms, today we are going to study the microbial life. See, our CSIR exam is very much interested in microbial growth, microbial diversity, microbial structure, and microbial genetics, microbial molecular mechanisms. All these areas are very much important in our CSIR exam. So we cannot just skip these portions and attend the exam. So we will begin with, as usual, we know that we always start microbial area with bacteria. Okay. See, bacteria are a particular topic which we always study for different kinds of exams where biology is being asked. So let us start this particular unit. As usual, we are going to start with the very common topic, bacteria. So let us make this class a little more interesting. So I want to ask you one particular question. Just simple question. Why bacteria is considered prokaryote? Can you just comment few of the reasons why bacteria is considered as a prokaryotic organism? Anyone? Can you please comment why bacteria is considered a prokaryotic organism? This is a school level question. I hope you are all aware why they are considered as a prokaryotic organism. Can any of you comment your reasons for classifying bacteria as a prokaryotic organism? Yes, Divya. Very good, Divya. Very good. Divya has commented that bacteria does not possess a true nucleus. It's an absolutely wonderful point. These are something which we study right from our school days, right? So bacteria does not have a particular nucleus. Instead, they have a nucleoid structure. It is not absolutely a nucleus or a nucleus membrane-bound structure, but it is an area where the chromosomes are situated. We call it as nucleoid, right? Yes, Diniga is commenting absence of membrane-bound cell organelle. Very good, Diniga. We know that bacteria does not possess a membrane-bound cell organelle. They have ribosomes. You should not mistake the ribosomes as a membrane-bound organelle. Okay, ribosomes are not membrane-bound organelle. They are present in both eukaryotes and prokaryotes. What is meant by membrane-bound organelles? Can you quote some examples? These are mitochondria, endoplasmic reticulum, and stuff like that. That kind of organelles are absent in our dear bacteria. But they possess ribosomes. Ribosomes are not membrane-bound organelles. You have to keep that in mind. All right. So these are some of the reasons why we consider them as prokaryotes. And obviously, you have studied about bacterial ribosomes. They are 70 S ribosomes. S stands for Svedberg unit. In the case of eukaryotes, the ribosomes are 80 S ribosomes. Correct? Okay, friends. So let us start. So which are the common bacterial shapes? See, these are very simple area, but while you are preparing for CSIR exam, you have to make sure that your base, the foundation level is too strong. Otherwise, you cannot write CSIR exam. Some people think that you have to go after high-end depth questions. Yes, we have to definitely go after them, but you have to make your base or your foundation strong. So let us start. So the first shape which you have already studied is cocci or spherical bacteria. Cocci are roughly spherical in shape. See, roughly. You cannot say that it's a perfect sphere, but it is roughly spherical in shape. May occur singly or in characteristic arrangements. They can occur as a single existence or they can combine together to form different kind of structures. But their individual cell shape is roughly spherical. And the arrangement helps in bacterial identification. So how they are getting arranged? Are they arranged as a pair or are they arranged as a long chain? Depends upon these arrangements. We can further classify the cocci bacteria. So cocci bacteria are roughly spherical in shape. It is a very simple point. So what are the different kinds of shape? One is diplococci. Friends, diplo means what? Di. Di means what? Di means two, right? Mono means one. Di means two. Tri means three. So diplococci means two cocci bacteria or two coccus bacteria. Cocci is plural, coccus is singular. So two coccus bacteria, if they are occurring in pairs, we call it as diplococci, right? Simple. So formed when two cocci remain attached after division. See, they have divided, one cocci has already divided, but they are not getting separated. Instead, they are attached to each other. So this will be the structure. They are not getting separated, but they are attached to each other, right? And they occur as pairs. Obviously, di means they will be in pairs. Another type of arrangement is streptococci. Okay, streptococci. See, I always tell my students to understand this term as strepto means strip of a medicine or strip of a tablet. You must have purchased several medicines from the medical store and they will give you a strip of particular capsules or tablets. So strip means a chain, something which is long. So a chain of cocci bacteria is called as streptococci. Cells divide repeatedly in one plane and form long chains. So this is how streptococci bacteria will occur. See, the overall structure is similar to a chain, but it is made up of individual cocci bacteria. Simple concept. So what are the examples of streptococci? Streptococcus is an example, enterococcus is an example, lactococcus is an example. See, always study examples for CSIR exams. Okay, CSIR exams requires very much knowledge about examples and how they work. So you have to study examples. So streptococci examples are streptococcus, enterococcus, lactococcus. These are all examples of streptococci. So strepto means in long chain. Simple area. There is no much confusion in this area. You can easily read and understand these topics. Coming back to the third one, it's staphylococcus. What is staphylococcus? Staphylococcus means arranged in grape-like clusters. See, this is one coccus bacteria. Okay. See, if the coccus bacteria is getting divided in a single plane like this, then definitely this will result in a chain, which is called streptococci, right? But just imagine, what if this particular cocci bacteria is getting divided in different planes? Okay, imagine I have a knife in my hand and I am just cutting it across different planes, like this, like this, like this, like this, like this. It will become a mess, right? So what is the result? See, we will get several cocci bacteria clustered like this, which gives an appearance of grape cluster. Grapes, you know what or how a grape cluster looks like. So this is how the staphylococci exist. There are several cocci in this existence, but why this is achieving this particular structure? Because they are getting divided in random planes. So what resulted in streptococci? The division was in one particular plane. That is the reason why we got a long chain-like appearance. But the division, if the division is happening across different planes or in random planes, we will not get a very nice ordered chain-like structure. This will result in a clustered cocci structure, and this we call it as staphylococci, and it is having a grape-like appearance. Example is staphylococcus. Simple. Staphylococcus. Okay. So this is staphylococci, right? Another one is tetrad. Friends, what is tetrad? Tetrad means 4 cells. Okay. So in the case of tetrad, the cocci bacteria will be existing in 4-cell stage. Okay. So if they have to exist in 4-cell stage, then you have to understand that the division along one plane won't give rise to tetrad. Okay. It will move towards a streptococci appearance. But if a cocci bacteria is getting divided in 2 different planes, see, imagine like this and like this, we will get 4 cocci bacteria. All right. So this appearance is called as tetrad. Some cocci bacteria exist in this particular form. What is the example? Micrococcus. Friends, please keep in mind, Micrococcus is an example of tetrad type of cocci bacteria. Tetrad means 4. And how the tetrad is getting resulted? One cocci bacteria is getting divided in 2 planes. That is how we get tetrad. Okay. So tetrad, an example of tetrad bacteria is Micrococcus. Simple concept. All right. And another different type of cocci bacteria is Sarcinae. What is Sarcinae? Division occurs in 3 planes. Very, very important. Please study that when a cocci bacteria is getting divided in 3 planes, obviously first division will result in 2 cells, second division will result in 4 cells, third division will obviously result in 8 cells. Okay. So if a particular cocci is getting divided in 3 planes, then what will happen? It will result in an 8-cell stage, right? So that is called Sarcinae. Very, very important. Very, very important. See, we always study cocci, rod-shaped bacteria like Bacillus, Vibrio, etc. Then we will close our book. But no, there are other kinds of shapes like tetrad, Sarcinae, which can be asked in our CSIR exam. So what is Sarcinae? You have to compare tetrad and Sarcinae. Tetrad will give rise to 4 cells, and the reason is division is occurring across 2 planes. What about Sarcinae? This will result in 8 cells. Okay. So the cocci bacteria is existing in an 8-celled stage, and this is the result of divisions across 3 planes. Tetrad was occurring in 2 planes. Sarcinae was occurring in 3 planes. Right. Example is Sarcina. All right. Example is Sarcina. See, examples are just the names of the types. All right. So we have studied what Diplococci is. We have studied what Streptococci is. We have seen Staphylococci. We have seen tetrad and Sarcinae. All right. Got it? Yes. So these are different types of cocci bacteria. I hope you have understood this particular area, right? So these are very simple area. And coming to the next shape, these are rod-shaped bacteria called bacilli. Bacilli is plural, bacillus is singular. See, bacilli is a very common term in microbiology. So these are cylindrical bacteria. See, cylindrical means rod-shaped structure. Okay, so this can be considered as a cylindrical structure, and either the question will be asked like rod-shaped bacteria or cylindrical bacteria. Usually CSIR exam won't prefer such kind of a simple type of question, but since as we are focusing on the basics, you have to understand cylindrical or rod-shaped or elongated. It can be asked in different types or methods. So what is an example of bacilli? Escherichia coli. Escherichia coli, the famous E. coli. It is an example of bacillus type bacteria. Legionella pneumophila, a different example of bacillus. Legionella pneumophila. This is also an example of bacilli. Okay, you can get n number of examples. I'm just focusing on 2 examples, right? So bacilli bacteria examples E. coli and Legionella pneumophila. What are their characteristic features? They vary greatly in length to width ratio. See, what does this statement mean? They can extend or their length can vary and their width can vary. So the length to width ratio need not be always same. They can vary. Okay. And they may occur singly or in pairs or in chains. Like cocci bacteria, bacilli bacteria can also exist single-handedly or as a single organism, or they can occur in pairs, or they can occur in chains. They can also do all what cocci is also doing. So these are rod-shaped bacteria called bacilli. And they are rod-shaped, but what about their ends? What about their end portion? Will they always be like a rounded structure, or can they become pointed, or can they become branched? Is it possible? Yes, it is possible. The bacilli bacteria's ends can be rounded, flat, football shaped, bifurcated. See, they can exist in different shapes. The end points of the bacilli bacteria can be rounded, they can be flat, they can be football shaped, they can be bifurcated. Bifurcated means somewhat like branched. All right. So these are all different types of existence of bacilli bacteria. So here we are focusing on the end portions of the bacilli bacteria. All right. Yes. So I hope you are clear about bacilli bacteria. Very, very simple topic. And let's see what is a cocci. We have studied what round-shaped bacteria are, cocci. We have studied what rod-shaped bacteria are. Now we are seeing a different word which is called coccobacilli. Oh, is that possible? Is that possible? Coccobacilli. Let's see. They are short and broad rods. Okay. See, it will exist somewhat like this. When we say this, we cannot make or we cannot understand what its actual structure is. Is it a rod? Is it a spherical shaped structure? We have no idea. Actually, this is a coccobacilli. Okay. Coccobacilli means they are actually rod-shaped bacilli bacteria, but they are short and broad. This shortness and their increased breadth gives them an appearance of a rounded structure, but actually they are a rod-shaped bacteria. So a question can be asked from this area. Coccobacilli is a coccus bacteria or coccobacilli is a rod-shaped bacteria? Okay. So it is a very nice question. Is coccobacilli a rod-shaped bacteria or is it actually a spherical-shaped bacteria? Some students will get negative marks if they try to attempt this particular question. So you have to keep in mind, coccobacilli is always a bacilli, which means they are actually rod-shaped bacilli bacteria, but unfortunately their appearance is somewhat similar to cocci bacteria. So that is why they are getting called coccobacilli. All right. They resemble the cocci bacteria and they form chains, or they are chain forming bacilli. So they usually exist in chains. Okay. So these are coccobacilli. Example, Bacillus megatherium. Please study this. Bacillus megatherium. What is Bacillus megatherium? It is an example of coccobacilli. What is coccobacilli? Is it a bacillus or is it a coccus? It is definitely a bacillus, but it is having an appearance like a coccus bacteria. All right. Example is Bacillus megatherium. Okay. Examples are really important. I hope you have understood this particular area. See, here we have beautiful images of different types of bacteria. Let's see. The first image is a cocci in chains. All right. A cocci bacteria in chains. Streptococcus agalactiae. This is a cocci bacteria in chains. See, cocci bacteria which is existing in chains, right? Second one, Staphylococcus aureus. Cocci bacteria seen in clusters. They are seen in clusters, not in chains. Third one, Legionella pneumophila. Legionella pneumophila. They are rod bacteria but existing in chains. Legionella pneumophila. Right? So these are several different kinds of existence of bacteria and their shapes, how they differ in shapes. And see, cocci bacteria and bacilli bacteria are actually really common in our nature. But there are some other types of shapes or some other shapes which are not that much common, but we have to study for our exam. Which are those less common bacterial shapes? One shape is Vibrio. See, Vibrio, comma-shaped bacteria, you have studied for several other exams. Vibrio means comma-shaped bacteria. Comma-shaped bacteria. It just resembles a comma shape. Comma-shaped bacteria. See, the famous example for Vibrio is Vibrio species. I mean, Vibrio cholerae. There are different types of Vibrio species. All right. Reason for cholera disease, Vibrio cholerae. So these are comma-shaped bacteria. Okay. Another type is Spirilla. See, the word suggests something. The shape is in a spiral arrangement. That is called Spirilla. See, Spirillas are rigid spiral-shaped bacteria. See, spiral-shaped bacteria are called Spirilla. But just look at this particular word. They are called spirochetes. They are also spiral-shaped bacteria. So Spirilla is also a spiral-shaped bacteria. Spirochetes are also a spiral-shaped bacteria. So what is the difference between Spirilla and spirochetes? Both are actually spiral-shaped bacteria. See, Spirilla is always a rigid spiral-shaped bacteria, while spirochete is always a flexible spiral-shaped bacteria. So that is the main distinguishing feature of Spirilla and spirochetes. Spirilla and spirochetes are both spiral-shaped bacteria, but Spirilla is a rigid-shaped bacteria or rigid bacteria. Spirochetes are flexible. They are not rigid like Spirilla. So that is the key distinguishing feature between Spirilla and spirochetes. And Spirilla often possess tufts of flagella. Tufts means groups of flagella. Spirilla usually possess tufts of flagella. And spirochetes, they possess internal flagella called axial filaments. This is very, very important point. Always getting asked in our exam. Okay. They possess internal flagella. The internal flagella can also be called as endoflagella. Okay. The actual name is axial filaments. So axial filaments are a characteristic feature of Spirilla or spirochetes. They are characteristic features of spirochetes. All right. So what is the difference between Spirilla and spirochete? Even if they are having a spiral-shaped structure, Spirilla is a rigid bacteria. Spirochete is a flexible bacteria. Spirilla usually possess tufts of flagella, which means groups of flagella, but spirochete is having endoflagella. Spirilla's flagellas are seen outside their body or their cell, but spirochetes flagella is an endoflagella or internal flagella, which is also called as axial filaments. Okay, and they belong to the phylum Spirochetes. Okay, clear? So some of the less common bacterial shapes are Vibrio, Spirilla, spirochetes. Clear? So we have studied what cocci is, we have studied what bacilli is, we have seen their different arrangements, and then we saw what Vibrio is, Spirilla is, and spirochete is. All right. Now let's see different other types of shapes. One is stalked bacteria. See, what is stalked bacteria? Stalked bacteria means they possess stalk-like extensions. From the bacterial cell, they possess extensions. These types are called as stalked bacteria. Example, Colobacter crescentus. Colobacter crescentus. See, what are stalked bacteria? Stalked bacteria possess stalk-like extensions from their body or from their cell. And their example is Colobacter crescentus. Okay. What about pleomorphic bacteria? See, pleomorphic means they can differ in their shape. They can differ in their shape. We cannot just assign one particular shape on them. Okay, they show variance in their shape. Pleomorphic means their shape is getting varied. We cannot just group them as one particular shape. So variable in shape, no single characteristic form is present in pleomorphic bacteria. So these are pleomorphic bacteria. So what is a stalk bacteria? Bacteria that possess stalk-like extensions. What is a pleomorphic bacteria? Bacteria which cannot be classified as a one particular shaped bacteria. They will always show variations in their shapes. These are called pleomorphic bacteria. Now let's come to multicellular-like bacteria. See, multicellular-like bacteria. We know that bacteria are unicellular organisms. That is why we call this as multicellular-like bacteria. The appearance is just like a multicellular organism, but they are actually unicellular. Filamentous actinobacteria. They form long filaments called hyphae. Hyphae collectively form a mycelium and they resemble filamentous fungi. See, you have studied what fungi is. You have studied how a fungal structure resembles or how it is actually arranged. They possess hyphae and all these hyphae together form mycelium. Okay. So similarly, these filamentous actinobacteria, they're actually bacteria, but they form hyphal-like structures and they form mycelium-like structures. So this will give them a fungi-like appearance. That is why we call it as a multicellular-like bacteria, even though they are not actually multicellular. Okay, they form hyphae or mycelium-like structures. So these are filamentous actinobacteria. Then there is filamentous cyanobacteria. See, cyanobacteria means they are able for photosynthesis. They are photosynthetic bacteria. They form long filaments and they show cellular differentiation. See, cellular differentiation means the entire group, they will be having different types of cells and each cells will be differentiated. Okay, somewhat like division of labor, we can call it like division of labor. So different cells will be doing different tasks. All right. So this type of differentiation, cellular differentiation, will give them an appearance of multicellular organism. Okay, that is why they are called as multicellular-like bacteria. So some bacteria can resemble a multicellular-like form. Example, filamentous actinobacteria and filamentous cyanobacteria. All right. So these are some of the different types of existence which we need to understand for our exam. All right. So we have gone through several types of bacteria and we haven't completed all these categories yet. So let's see. Yes, here you can see different types of shapes or different kinds of bacteria which we have discussed. So the first one is a comma-shaped Vibrio bacteria. You can cleanly observe that the shape is just like a comma. All right. It is very evident from this picture. The second picture is a spiral-shaped bacteria. See, they are spiral in structure, but they are rigid. All right, they are rigid. They are spiral-shaped bacteria. Okay. Third one is a spirochetes. See, spirochetes, they are a little bit longer and they are more flexible than the spirella bacteria. Okay. Now let's come to the stalked bacterium. See, stalked bacteria, you can see there are stalk-like extensions from their cells. All right. There are stalk-like extensions. See, these are all stalk-like extensions from their cells or from their cell body. These are called stalked bacterium. Okay. Now here you can see filamentous bacterium. Filamentous bacterium. The bacteria is arranged in a filamentous form, giving it an appearance of a multicellular organism. These are filamentous bacteria. And here you can see bacteria in the form of a fruiting body resembling fungi. They are getting into the formation of a fruiting body. So these are all giving an appearance of a multicellular organism, but in fact they are actually multicellular-like bacteria. Okay. Now I think the structures are clear. And now let's come to a different word. This is called heterocyst. See, have you heard about the word heterocyst? Some of you must have learned what heterocyst is. These are specialized cells which can perform nitrogen fixation. Okay. When there is a long chain of bacterial cells. Some cells may get converted to heterocysts, which have a sole function to perform nitrogen fixation. We study heterocysts in different microbial areas where we focus how heterocyst helps in nitrogen fixation. But right now, just keep in mind that heterocysts are specialized cells which do nitrogen fixation. All right, this is a very important term. This is a very important term, heterocyst. So what are heterocysts? They are specialized cells dedicated to perform nitrogen fixation. So nitrogen fixation will be efficiently performed by heterocysts. So these heterocysts are occurring in different types of microbial life. Now, what is a myxobacteria? Myxo, it is an aggregate to form multicellular structures producing fruiting bodies. Right now, we have seen such kind of a picture where the bacteria are getting aggregated to form a fruiting body. So that type of bacteria can be called as myxobacteria. They aggregate to form multicellular structures and they produce fruiting bodies. Okay, let's go back to that particular picture. See, here you can see how that particular bacteria is forming a fruiting body, right? They are getting aggregated to give a multicellular like appearance and they resemble fruiting body, which is something which we will study in the fungus or the fungal portion. So these are called myxobacteria. Myxobacteria. All right, myxobacteria. So there are different types of structural existence for these bacteria. Okay, so this is what we should study or which we should be aware of when we start the bacterial portion. Okay, we will always study what coccuses, we will always study what diploid or the bacilli is, but you have to understand there are also different types of shapes. All right, can you please comment if you are with me right now? You understood what the different shapes of bacteria is? Can you please give a comment so that I can understand that you got this topic? Is it clear? These are very simple areas which you can just read and understand. Okay, but you have to cover these particular points also. Yes, friends, I think it is clear. There is no doubt in this particular topic. Okay, friends. So let's move to a different area. Different means something related to bacteria, but a different under a different heading. Yes, Divya, clear. It is clear. Okay, okay. So now let's move on to the size of bacteria. See, you can only say an average size for bacteria because science is evolving. The microscope is also getting evolved. So different types of bacteria which were not earlier identified are getting identified right now. So you can only study an average size of the bacteria. Not even bacteria, even any microorganisms, you can only take average size. Okay. So what is the average bacterial size? See, we always take E. coli as a reference model organism. That's why we are usually seeing the word E. coli in different scenarios because it is a model organism which we take in the inside the microbiology topics. See, we consider E. coli as a model organism and let us see what its width is 1.1 to 1.5 micrometer. Its length is 2 to 6 micrometer. All right, for CSIR exam, you have to study the dimensions or the length and the width. You have to have an understanding of what is the average size of a bacteria. This can vary depending upon the bacterial shape. Obviously, a cocci bacteria or a coccus bacteria won't be having that much kind of length and all, but definitely these values are an average value which you should keep in mind. Okay, so E. coli is usually taken as a model bacteria. Okay, so their width is 1.1 to 1.5 micrometer and their length is 2 to 6 micrometer. All right, now what is the smallest bacteria? Which one is the smallest bacteria? It is the mycoplasma, or also called as PPLO, pneumonia-like organisms. These are school level points. You must have already heard about all these things. Mycoplasma, they are also called as PPLO. So these are the smallest bacteria. Actually, there are other new discoveries related to the smallest and largest, but let's stick on to the bookish information that mycoplasma is the smallest bacteria, right? And their approximate diameter is 0.3 micrometer. So the mycoplasma, which is actually a bacteria which does not possess a cell wall, which we will study later. And this particular mycoplasma, their diameter is 0.3 micrometer. You should study this diameter. Since mycoplasma is something which is having an academic value, you have to study what their diameter is. Their diameter is 0.3 micrometer. All right, clear? So please keep this in mind. Mycoplasma's diameter is 0.3 micrometer. Okay. So we saw what or which one is the smallest bacteria, but can we classify the largest bacteria? So these are areas where active research is going on. Different bacteria are getting discovered. So let's see which are some of the largest bacteria. One is spirochetes. See, spirochetes are actually, you saw the diagram. Spirochetes are very much lengthier bacteria. So their length can range up to 500 micrometers. See, 500 micrometers for a bacteria, it is pretty large. Their length is pretty large, or they are long enough to be classified as large bacteria. And oscillatoria, this is also a large bacteria where their diameter is 7 micrometer. See, mycoplasma was having diameter 0.3 micrometer, but here oscillatoria is having a diameter of 7 micrometer. You have to compare how much is 0.3 and how much is 7. So this is the difference. So oscillatoria is one of the largest bacteria with 7 micrometer in diameter. And they're similar to the diameter of red blood cells. See, there are bacteria like oscillatoria which possess diameter which is almost equal to red blood cells. Red blood cells are almost having this diameter. Okay, so they are as big as a red blood cells or RBC. Another large bacteria is Epilopisium fischelsoni. Epilopisium fischelsoni. This is also a large bacteria, and their size is 600 to 80 micrometer. Their dimensions are 600 to 80 micrometer, and they are larger than many eukaryotic cells. Please study this. Please keep this point in mind. Which among the following bacteria is considered as larger than most of the eukaryotic bacteria or eukaryotic cell? Not eukaryotic bacteria, eukaryotic cell. That is Epilopisium fischelsoni. Very important. You may find it a little bit difficult to study this name, but it is actually important. It is a type of a large bacteria, okay, which is having a dimension of 600 to 80 micrometer, and they are larger than many eukaryotic cells also. Okay. Coming to the next one, Thiomargarita namibiensis, one of the largest known bacteria. Thiomargarita namibiensis. Okay, these are one of the largest known bacteria. Obviously, there are other bacteria also. So that is why it is written as one of the largest. Okay, so Thiomargarita namibiensis, these are one of the largest known bacteria, and they are found in marine sediments. Okay, so Thiomargarita namibiensis, these are seen in marine sediments. That is a very important point. See, in our CSIR exam, the question won't be a straight question. Okay, sometimes the question will be in such a manner that you have to pick certain examples from the question, or it will be in a match the following type. It can be put forward in different kinds of questions. Okay, so we always think that, okay, which is the largest one? So what is the dimension of this particular bacteria? The question won't be in such a manner. So you have to be prepared. Okay, so we are seeing some of the largest bacteria. Spirochetes are obviously large due to their length. Oscillatoria is a large bacteria. Epilopisium fischelsoni is a large bacteria which is actually larger than many eukaryotic cells. And Thiomargarita namibiensis is one of the largest known bacteria which are always found in marine sediments. Okay? Yes. So these are very important points. Yes. See, here is an image of Epilopisium fischelsoni. Okay? Epilopisium fischelsoni. Look how these bacteria are. See, from here we can't judge the actual the size of the bacteria, right? We just observe it as some sort of smaller things, but actually compared to the normal bacterial size, these are very much large. Okay, see these large bacteria. See, for comparison, you have Paramecia, which is a protozoa. You can see, I will use a different color. You can see Paramecia here. All right. See, Paramecia is a protozoa. So compared to the Paramecia, look how large these particular giant bacteriums are. All right. So this is for your comparison. This is for you to understand how big this particular Epilopisium fischelsoni is. So compared to these protozoa, Paramecia, these giant bacteria are actually, they are looking so huge. All right. And the microscope which is used to identify this is phase contrast microscope. All right. See, when you are preparing for CSIR exams, please make sure that you are reading the reference text. Please make sure that you are not skipping the diagrams and images. In the case of reference text, every points are not given as text. All right. They may be included under the description of images. That is why, that is the single most reason why we always ask our students to read the reference text without skipping the pictures. Okay. So from this picture, it is clear that for observing these kinds of bacteria, we normally use phase contrast microscope. So this can be a question. Which among the following microscopes are used to compare the sizes of bacteria or to visualize the bacterial size? So these can all be asked as a question. So we will get the answer only if we read the pictures from the reference text. Okay. So this is a giant bacterium and this is how it is looking against the protozoa Paramecia. Now, what is the importance of bacterial size and shape? Okay, these are not just simple introductions to the bacterial portion, but there is a reason why we study the size and shape of the bacteria. So what is the importance of the size and shape of the bacteria? Let's see. Surface area to volume ratio. Okay. See, the surface area of the bacteria when compared to its volume, it is a very important parameter. Okay, why is it important? Let's see. The higher surface area to volume ratio, the nutrient uptake will get improved, waste removal will get improved, diffusion efficiency will be high, growth rate will also be high. Okay, this is a very important area for our exam. All right. Higher the surface area to volume ratio, the maximum nutrient uptake, waste removal, diffusion efficiency and growth rate will the bacteria show. See, just imagine the bacteria is having a particular volume and what if their surface area is getting higher? Just imagine how the intestinal villi inside our body is behaving. Why we possess intestinal villi? Intestinal villi is actually increasing the surface area so that maximum nutrients can be absorbed in our body. All right. So definitely when surface area is getting improved or higher compared to the volume, definitely the these kind of process will, their efficiency will increase. So that is the single most reason why our intestine possess villi, and inside that villi there is also microvilli. So again the surface area is getting improved. So that is why if the surface area to volume ratio, so you have to understand this is always taken in account with the volume. See, if volume is also higher compared to surface area, then the actual value won't change. So compared to the volume, the surface area should increase. Okay. Otherwise, we can just say that if surface area is high, then all these processes will also be higher. No. The surface area should be higher compared to the volumes, which means the volume should somewhat remain constant. Only in that case, all these processes will get increased. So why we are studying about the bacterial size and shape? Then we will get an understanding about what is their surface area to volume ratio is. If the surface area to volume ratio is improving or it is getting higher, then definitely the cell's nutrient uptake, waste removal, diffusion efficiency and growth rate will improve. So this is a very, very important section which you should keep in your mind. All right. Simple point, simple area. So what is the influence of shape? Rod-shaped bacteria have a higher surface to volume ratio than cocci of equal volume. Students, please keep this point in mind. This can be asked for our exam. Which among the following statements are correct? Which among the following statement about the bacterial shape is incorrect? This is a very important point. Rod-shaped bacteria have higher surface to volume ratio, surface area to volume ratio than cocci of equal volume. If we compare a bacilli and if we compare a cocci bacteria, if both of these bacteria are having constant volume, then definitely the rod-shaped bacilli will be having higher surface area. Okay, so this is a very, very important statement. If we keep the volume constant, then definitely the rod-shaped bacteria or the bacilli will show higher surface area ratio than cocci bacteria. Then it can also be interpreted in this manner that the rod-shaped bacteria will be having higher nutrient uptake efficiency when compared to the coccus bacteria of the same volume. Definitely that can also be understood, right? So this is a very important statement. Please keep this in mind. And rods can absorb nutrients more efficiently. Yes. Another point which can be asked in our exam. Rod-shaped bacteria can absorb nutrients more efficiently. See, when we start studying microbiology or microbes for our CSIR exam, we always focus on their genetics. We always focus on their molecular biology. We just ignore the shape and size part of our microbiology or bacteria. See, why are we studying all these things? If we get a statement in our exam that rods can absorb nutrients more efficiently, we will always think about, if this is the case, then there should be different types of absorbing carrier molecules or channels. We will be thinking in that way. But this has got nothing to do with carrier molecules or channels. This is purely related to their surface area to volume ratio. All right. So why rod bacteria are getting more efficiency for nutrient uptake? It is because of a simple reason. Because rod-shaped bacteria have more surface area to volume ratio than cocci bacteria. Simple. Now, what are the adaptations of larger bacteria? Large bacteria, obviously due to their size, they require special adaptations. Okay. Example, Epilopisium fischelsoni has a highly convoluted plasma membrane, and this will increase the surface area. Okay. See, just imagine you are traveling in a bus. Okay. You are traveling in a bus. And just imagine that you are a big guy, or you are having so much of fat in your body. Just imagine that you are a fat person and you don't want to disturb other people sitting near your seat. So how can you occupy that seat without disturbing them? You will always try to shrug to yourself, right? You cannot just go and sit in the seat like this. Okay, that will definitely disturb the other passengers. So you will always try to shrug, or you will always try to make your body a little bit smaller, okay, in order to accommodate in that particular seat. So this is also the similar case with these larger bacteriums, because they cannot just exist in their actual shape. Their plasma membrane will definitely get convoluted What is convoluted? Convoluted means somewhat twisted, somewhat complex structure, because they want to get accommodated in their specific area. All right. So they have these kind of adaptations where the larger bacteriums like Epilopicium, Fischelsoni, they has a highly convoluted plasma membrane, and definitely this will increase their surface area. Obviously, that will increase their surface area. So these are some of the adaptations shown by the larger bacterium. Okay, simple portion. And there will be a protection from predation. Let's see how this is happening. Large bacteria are less likely to be eaten by protists. Filamentous, stalked, and unusual shapes also reduce predation risk. See, due to their massive size, the protists, which usually depends upon the bacterium, or which usually feed on the bacterium, due to this larger size of the bacterium, most of the protists won't attack the large bacteria. Okay, they will always go behind traditional bacteria or normal shaped bacteria, and the larger size of these bacterium will help them to avoid the predators like protists. Okay, and their filamentous, stalked, and unusual shapes. These unusual shapes will make the protist to go after normal shaped bacteria. Okay, they won't feel like, they are not feeling this larger bacterium as if they are normal bacteria. Okay, protista will come and see, oh, okay, this is a different type of structure. I don't think these are bacteriums, or I don't think these are typical bacteria, so I will go after our usual bacteria. So that is how they avoid predators like protists. So the larger size of the bacteria, they are showing different types of adaptations, and they will definitely help in avoiding predations. Okay, so please keep this point in mind. All right, friends, so we have studied what the size and shapes of bacteria are. We have already seen how they are helping in their absorption of nutrients, how they help in their larger bacteria, how they are getting benefited from their size to avoid predations. We have already seen all these stuffs. So today's class, we have focused purely on size, shape, and their importance. Okay, so now we have to go in detail about the different structures inside and outside a bacterium, so that you can get a proper understanding about the bacterial cell. From these areas, different types of questions are getting asked for our CSIR exam. Okay, regarding plasma membrane, regarding cell wall, how materials are getting transported, different types of transporters. All right, that all we will study in our next class. Okay, so today we have concentrated on the size and shape of the bacteria and the different importance of their surface area, volume ratio, and adaptations of their larger size, all those things. Next class, we will purely concentrate on the different structures of the bacteria. See, you can see bacteria possess capsule-like structure, ribosomes are seen inside the bacteria, cell wall is seen, plasma membrane is, you can see plasma membrane, they are possessing fimbriaceous structures, chromosome DNA, right? This is not a nucleus. The chromosomal DNA is seen in a particular area called nucleoid. Okay, and they possess flagellum. We have to study in detail how a flagellum works, how a prokaryotic flagellum is different from eukaryotic flagellum. These are all hot topics for CSIR exam. And there are several inclusion bodies, different types of inclusion bodies, how they work, what they are getting, which components are getting stored inside the inclusion bodies. We have to concentrate on all those things. Okay, so friends, I think you are happy with this particular area. I think you have understood the concept of different size and shapes of bacteria. So can we conclude today's class? Okay, I think the size and shape of bacteria, that portion is clear to you. Okay, friends, so together let us crack this exam. So there is a systematic way to study for CSIR exam. You have to understand the basics, and you have to read the reference text. That is a very mandatory thing to do while you are preparing for CSIR exam. You have to read in depth the reference text, and then you have to attempt several MCQs related to the exam. So this is how we crack this particular exam. All right. Okay, friends. So we can conclude today's session. I hope you understood what we have discussed here. So we will see in our next session. So where we will concentrate on the structures both inside and outside a bacterial cell. So thank you so much. Have a nice day.